Greetings and welcome to this module on plant molecular genetics. In this module, we will be discussing the genetic engineering in terms of the application of gene constructs to modify plants. The objective of molecular breeding is to develop plant varieties with desired traits. Unlike conventional breeding, which is founded on the selection on the basis of phenotypes only, molecular breeding is based on the genetic engineering of plants with specific genes which will result in the desired phenotype. What one needs to understand is that genetic engineering is basically a very specific process which involves identification of specific traits or genes linked to specific traits and transference of these genes into the host plant, which in turn will manifest that particular trait in the phenotype. So this is a basic example of what happens in transgenesis. So we have a plant which is the transformation variety. In this case, it can be a selected hybrid variety or a conventional plant which may not have a desired characteristic. This is the transgenic construct. In some cases, these constructs may encode genes which confer additional resistance to pests or maybe aroma or other desired traits, and this gene is transformed into this variety. Unlike conventional breeding, which relies on two plants or the two parental genotypes to produce the F1 hybrid, genetic engineering relies on the introduction of a construct. So we have only one plant. We then proceed through 3 generations of self-fertilization in order to stabilize the gene. And what one needs to note is that genes may be lost because these are artificial constructs, and they may be lost as a result of genetic drift or also as a result of genetic recombination. And finally, you have your transform variety. So once you have a transform variety, one needs to crossbreed this with an elite variety, or what is known as a commercially utilized strain, and that backcross results in the transformants. Now at each stage of this process, the breeder needs to track these genes using molecular markers, which I have discussed in my earlier lecture. These molecular markers can be tracked using PCR-based methods. Let us look at an overview of the steps in molecular breeding. So the first step is obviously the identification of the desired traits. So these traits can be those which are desired commercially, such as the increased tolerance to pests, more response to fertilizer, what is known as an elevated response to fertilizer application, as well as the traits such as yield. So these are the desired traits. The next path or the next step will involve the characterization of the pathway, identification of genes involved in the pathway because most traits are polygenic, isolation of these genes, development of the construct, transformation of the target plant with the construct using any one of the delivery methods, which may be biolistic or agrobacterium mediated. Then there is transformation, the actual transformation of the host plant, and finally screening and commercialization. So this gives an overview of the entire process of genetic engineering. Let us look at a case study. So in this case, we are looking at a single gene which encodes a particular trait. Okay, so this is a protein which will confer drought tolerance in Oryza sativa. However, this protein is only found in the wild type variety of Oryza sativa, but the wild type is not fit for commercial breeding because it has undesired characteristics. Now, as a breeder, you want to transfer the genes encoding this particular protein into an elite variety of Oryza sativa which does not have the characteristic of drought tolerance. Let us see how we proceed. So the first step will involve characterization of the pathway. So this is the end product of that pathway in the wild variety. However, this protein DRR is not the product of a single gene. Rather, it's a product of multiple genes operating together in a pathway. So it starts off with product DRA, then DRB, DRC, DRD, and finally DRR. So each step of this pathway is basically catalyzed by one gene. Okay. So we have 1, 2, 3, and 4 genes. These 4 genes work on the subsequent substrates to result in this product. Now what one needs to do as a genetic engineer is identify each of these particular enzymes. And once you have identified these particular enzymes, you can move on to the development of your construct. So remember, this is a trait which is polygenic trait, and one needs to have all these 4 genes transformed into the host plant in order to manifest this particular phenotype. So transformation and selection is not that easy because each of these particular genes may be located on different chromosomes in the wild type. So you have these examples. You have DRase1 located on chromosome 1, DRbase2 located on chromosome 3, and so on and so forth. Now one needs to isolate these particular genes. Now take note that each gene may be containing its own specific promoter and the introns, which in turn may determine the level of expression. So one needs to isolate each of these genes using PCR, sequence them, and characterize them. So we have isolated all of these 4 genes, and now we intend to develop the construct. So in order for these gene construct to be transcribed and translated in the host plant or the target plant, one needs to attach a promoter, and the selection of this promoter is based on the host plant. One needs to use a promoter from that host plant. Now the selection of promoter can also be done using a promoter which will overexpress, such as the 35S promoter. However, overexpression may not result in the ideal phenotype, as these genes or their transcripts may interfere with other genes and result in other complications such as the gene silencing or the RNA interference. So these are factors which one needs to take into account when designing gene constructs. So we move on to the construct. So this is a theoretical construct. So I am going to use agrobacterium, and this is the plasmid from agrobacterium, and you have a recombination site. These 2 are recombination sites which will mediate the integration of these constructs. So this is the construct itself into the host plant. In addition to this, we will also have the reporter genes. The reporter gene which may result in a fluorescent signal and which will enable the researcher to track the expression of this particular construct or to verify the integration of this construct. You may also have a selectable marker, which in this case can be resistance to a specific antibiotic. So this is a theoretical gene construct. If one examines the constructs which are used for agrobacterium, they will be much more complex, but I have simplified this for your reference. So once you have your construct ready, you can transform it using any of the methods. You can use biolystic methods, what is known as the gene gun, in which case your genes are coated onto nanoparticles of gold and then ballistically introduced into the plant using a high pressure volume of air or nitrogen. Or you can also use cauliflower mosaic virus mediated transformation or the conventional agrobacterium mediated transformation. So this selection of the method is based on your specific laboratory equipment. Okay, so once you have transformed your gene, you usually begin with a callus, the callus tissue, and the genes are transformed into this callus. So you do a biolystic transformation of the callus. Subsequent to this, you screen the callus using the selective marker. Usually use hygromycin or neomycin as a screening medium, and you select transformants. Now you get regenerants, but one of the things which you need to take into account is that some of the regenerants may be chimera. Chimera means a part of it is expressing the gene of interest and a part of it is basically the original plant or the original callus. So one needs to be careful when selecting regenerants. Usually you use a higher concentration of the hygromycin or the selecting agent in order to isolate more stringent isolation procedure for the transformants. Then you get shoot regeneration, which you can induce in tissue culture using a phytohormone combination. You induce the roots, and finally you get the grow out and the harvesting of seeds. This is the basic procedure for the transformation of plants. Now, when you select genetically engineered plants, you need to self them so that the plants are self-fertilized and you get the seeds which are true to type. If you conduct these kind of experiments in an open breeding environment outside the greenhouse, there's a high likelihood of getting cross-pollination. So these are some of the points which you need to consider in this particular system. And finally, you screen for transformants. Now, screening for transformants can be done using the DNA-based method, which I have mentioned in my earlier lecture, which uses PCR-based markers, or you can also screen for specific enzymes or hormones which are produced in the plant in response to that particular gene. So in this case, we have transferred a particular protein, so you can develop basically antibody markers for that particular protein and screen the plants for these particular antibody based markers. Okay, so what can go wrong when a foreign gene is introduced into a plant? Now, one must remember, based on the laws of selection, a foreign gene is a foreign object into the plant. So the plant's host machinery, which is the genetic machinery, will recognize this as a foreign gene. So the gene may not integrate into the host genome. So it may express transiently. Now please take note that when you use agrobacterium mediated transformation, you may have multiple integration events or none, as the integration points may be at multiple locations on multiple chromosomes. So if you have multiple integration events, you'll have a higher gene dosage and you'll have an overexpression of the gene. Sometimes genes may express transiently or not at all. So this is in turn governed by certain processes in the plant genome, such as the RNA interference as well as the recombination events which may occur over successive generations. Sometimes the gene may be integrated but not expressed, and this is another factor which is promoter driven or may also be driven by the exclusion of introns in the the genes. So these introns sometimes may encode what are known as the MIR trons or the microRNA introns, and removal of these introns in order to facilitate efficient gene expression may not be a good approach in many genetic engineering experiments. It would be better to leave the intron as it is and amplify the whole gene along with the introns and exons and introduce this into the plant, as the introns may contain regulatory circuits. Okay, and the third point is the gene may only be expressed in the first generation. Now this is ideal for companies which are involved in the sale of seeds, as they can continuously market new seeds, but not good in the case of plants which are developed for seed, for farmers to develop seeds. So this is basically a trait which is desired by genetic engineering companies. However, it's not desired in conventional breeding, in which case your seed becomes sterile and you can no longer use that particular seed. So what makes genetic engineering difficult is basically the fate of the RNA. Once the RNA transcript is transcribed in the plant, one does not know the fate of the transcript, as multiple processes may work together to basically limit this transcript. The other one is promoter functionality. Plants contain thousands of promoters, and the selection of the appropriate promoter is dependent on that particular plant, or it may also be linked to tissues. So specific promoters may be located in specific tissues, also located at different points in the plant, as well as in different times of the plant growth cycle. Some promoters may not express during growth, they may express during flowering. Some promoters may be turned on during fruiting and things of that nature. There's a loss of introduced genes, sometimes due to recombination events. If genes are not tightly linked, they can basically be dispersed and lost. There are also what are known as lethal introductions, or genes which can encode proteins that may, for instance, produce allergic symptoms in consumers, and these are lethal introductions. And the other one is interference in regulatory pathways. Overexpression of genes in plants may lead to the disruption of other regulatory pathways via allosteric inhibition. So these are factors which are far beyond the control of genetic engineers. As more and more genomes become available and more information becomes available, we may have greater control or greater insight into these factors. But currently, we have to rely on trial and error for gene expression. So this is a allosteric inhibition. So you can have a molecule which binds to an enzyme, which is a product of another genetic pathway, and then turns off this enzyme. These are factors which will affect your genetic engineering experiment. So one of the factors which you need to consider in terms of biosafety and biosecurity is the transgene escape. So you have transgene escape, in which you have your GM corn, and then you have local land races of corn or local varieties of corn, as has been reported in papers from South America. The genetically modified corn DNA has been found in local land races of corn, and this has in turn led to repercussions for local farmers. So these are factors which you need to consider when you develop genetically modified plants. Ideally, the genetically modified plants must be sterile, or else you will have a transgene escape. The pollen goes into the local land races, and then you have basically polluted the landscape with engineered genes. Okay, so one of the concepts which you must consider is the concept of linkage drag. I have put in a question bank, and I have also posed a question for you in this particular module. If you were to choose between conventional breeding and molecular breeding, which one would you select and why? So that brings us to the end of this module. Thank you very much for participating in this module, and I wish you a happy learning experience.